Hello, dear students. In last lecture, we have discussed about the different methods of classification and how we can classify the microorganisms into different kingdom. So, in this lecture, we'll discuss about the what are the different major characteristics which are used in the taxonomy so that we can place or we can identify that particular organism with respect to their different characteristics. So here we'll discuss one by one. The identification of an organism is the process of determining its species. So when we talk about the identification, we will use different methods with respect to their morphological characteristics, physiological characteristics, biochemical characteristics, and on that basis, we can place that particular organism into a particular group. So by this way, we can reach up to the species level so that we can clearly identify that whether that organism is already reported or whether it is newly isolated organism. And with respect to that, we can by following the particular rules, we can complete the nomenclature of that particular organism and place into a particular group. Various characteristics of an organism are determined by appropriate observation and taste, and these are then compared with the published description of the various species. For identification of microorganism, we can carry out different methods or different tests, or we can say different characteristics we can analyze in your laboratory so that we can reach a particular final conclusion so that our organism belong to a particular genus or a particular species. So what are the characteristics are used in classification system? We will discuss one by one. So when we carried out the proper protocol for the identification process, we will go through the different characteristics. Like we have to discuss about the what are the morphological characteristics of that particular organism, what are their physiological characteristics or immunological characteristics. Again, we will or we have to discuss about their metabolic requirement or metabolic characteristics, the etiological characteristics, their protein composition, or we can see with respect to their cell wall composition. We can say also composition of nucleic acid, what type of nucleic acid they contain, their hybridization method, nucleic acid sequencing with respect to sectionless RNA sequencing. Uh, uh, by this method, we can uh, conclude a particular that organism whether that belong to a particular group or it is already reported, or we can say we reach up to a species level. So we'll discuss what are the morphological characteristics which are used in the classification of that particular organism. So here, determination of morphological feature usually require studying individual cells of a pure culture. So when we talk about the morphological characteristics, the first requirement for the morphological characteristics becomes we have to require that particular organism in a pure culture. The microorganisms are very small in size, usually expressed in a micrometer. So when we talk about the bacteria, their size are ranges or we can measure in a micrometer. So when talk about the morphological characteristics, we need a particular culture in a pure form, and with respect to that, the size, shape, and arrangement of the cells are determined by microscopic examination of the stain smear. So after isolating that organism in pure culture, we can carry out the different morphological characterization of that particular organism with respect to the size, shape, and arrangement of that cell. And by microscopic examination or by staining procedure, we can um, determine a particular organization or we can say arrangement of that particular cell. For that size, shape, and arrangement determination, we can use different staining method as well. So examination of living organism in a hanging drop preparation shows motility, or when we categorizing organism into gram positive or gram negative with respect to that, we have to stain that particular organism by following gram staining. You can say again acid fast staining, we can also say flagella staining. Okay, so these are the different staining procedure we can use for the proper identification of that organism or to study the morphological characteristics of that organism. Apart from that, to determine whether that organism is motile or not, we carried out the process or the follow the process of hanging drop preparation, or we can say hanging drop method, to determine the motility of that organism. Are differentiated on the basis of various staining techniques, like frequently used staining methods are gram staining. On that basis, we can differentiate organism into whether they are gram positive or gram negative, in which the cell wall composition differentiate that organism into two groups, gram positive and gram negative. Acid fast staining, flagella staining, capsule stain, spore staining, and negative stainings. These are the different staining method we use for the differentiating that organism from one another. The gram staining is of great value because organisms are classified as gram positive or gram negative on the basis of that gram staining procedure. The staining reactions, however, vary with the composition of the medium, condition of growth, and the age of the culture. So these are the different affecting factor on which our result may be vary. So depending on the composition of that organism, that result can be we can observe. With respect to that, we can differentiate that organism into different groups. Now, when we stain the organism, we can observe that organism under microscope. With respect to that, we can determine or we can observe the shapes of bacteria. So bacteria have different shapes like foci, bacilli, vibrio shape, spirilla shape, filamentous, or mycoplasma. These are nothing but the different shapes of bacteria we can see under microscope. So what do you mean by foci? When we observe the bacterial cell, they are small in shape, spherical in shape, or oval in shape. So that bacterial cell we can call as that and nothing but the foci shape of that particular organism. And for example, on the foci shape we can observe uh, like micrococcus type of bacterial cell we stain, and we can observe under microscope. So micrococcus can observe in a foci shape always. Another shape is nothing but the bacilli, or we can also say call it as nothing but the rod shape. These are rod shape bacteria. And for example, rod shape bacteria is we are taken nothing but the bacillus, or the genus bacillus are nothing but the all are rod shape bacteria. Vibrio, it is again another shape of bacteria, or in the slide we can say it is nothing but the comma shape of that particular bacteria. They are comma shape, or we can say curved rod to that particular organism. And the example of comma shape bacteria or Vibrio organism are nothing but the Vibrio cholerae. So on the basis of their shape, we can differentiate that organism from one to another. Spirilla, it is nothing but the another shape of bacteria. They are longer rods. They are long from that of the rod shape of bacteria that we have discussed in bacillus shape. They are longer rods with several curves or coils, helical in shape. For example, Treponema pallidum, a causative agent of syphilis, is a spirillum shape of bacteria, or we can say this shape we can observe under microscope. Filamentous type of filamentous shape of bacteria we can also observe. They are branch-like filamentous bacteria, like in case of actinomycetes, we can see that type of structure. Mycoplasma, cell wall is absent in this bacteria. As micro, when we discuss about the mycoplasma, normally in their characteristics we can say they are cell wall lacking organism. Uh, hence, do not possess a stable morphology. They occurs round or oval in shape. So, the shape of that particular organism with respect to that. So, on these different shapes, we can differentiate that organism into different groups. Now, after when we observe the shape of bacteria, we can also see the proper arrangement pattern of that particular bacteria. As bacteria follows this special arrangement pattern, and on that basis, we can differentiate organism, or we can say bacteria, into different groups. Bacteria are typical, or the bacteria have a typical arrangement pattern. uh, their reproduction, or they reproduce by means of binary fission and split into a form in new cells. So in next lecture we'll also discuss about the what is meant by the binary fission. And bacteria are normally carried out the process of binary fission, or replicate by means of reproduce by means of binary fission. So Apart from that, they also follow the continuously proper pattern of arrangement. So when we see a diplococci type of diplococci shape, they are split along one plane and found arranged into pairs. So if we see if two cells are arranged into attached with one another, we can call it's nothing but the Diplococcus type of organism. For example, that type of arrangement we can see in case of Diplococcus pneumoniae. Streptococci, the strepto is nothing but the it is nothing but the arranged in a chain. So they divide on one plane and remain attached to form a chain. So this type of arrangement we can see in case of Streptococcus lactis. So it is again another type of arrangement of bacteria, as we have seen in case of size of shape of bacteria. Apart from that, bacteria also follow the proper arrangement pattern. Tetracocci, they are divided into two plane and live into group of fours. So tetra is nothing but the four, and if we see that particular bacteria into group of four, we can call it as nothing but the tetracocci. Staphylococci is nothing but they divide in three plane with respect to binary fission. How they follow the splitting around the plane with respect to that bacteria arrange into diplococci, streptococci, tetracocci, staphylococci. In case of Staphylococcus, they divide in three plane in irregular pattern. Looks and they look like a bunch of grapes. As we see the bunch of grapes, I like that type of arrangement we can see for the Staphylococcus aureus. So with respect to that, we can identify the particular organism, their shapes and arrangement pattern. Sarcina is again a divide in irregular pattern and produce cubical arrangement of group of 8 cells. So if we observe that type of arrangement of bacteria, we can say they are nothing but the Sarcina. While in case of bacilli, they split only across their short axis and group as a diplobacilli. For example, Klebsiella pneumoniae, Streptobacilli, example of Bacillus subtilis, these are nothing but the they split across their short axis. So with respect to that, their arrangement, they are differentiated into different groups. So here in this diagram, we can see a arrangement pattern of bacterial cell. In case of arrangement of cocci, if a single cocci we can found, it is nothing but the single, we can say cocci shape of bacteria. If that cocci are in a group of two, we can say it is nothing but the diplococci. It has nothing but the tetrad or tetracocci. We can say it is nothing but the Sarcina type of arrangement of bacterial cell. It is nothing but the Staphylococcus type of arrangement cell. It is nothing but the Streptococci. So if the cocci are arranged in a chain, we can say it is nothing but the Streptococci. With respect to the plane of reproduction, or you can say with respect to binary fission, they are arranged into particular manner. When we discuss about the arrangement of bacillus shape, single bacterial cell or rod shape bacteria, we can say it is nothing but the bacillus. When they found in two group of two cells, we can say it is nothing but the diplobacilli. And if we found that bacteria into a chains, we can call it as a streptobacilli. It is nothing but the palisade type of arrangement of bacterial cell, and it is nothing but the cocobacilli. In case of spirochete, spiral shape arrangement, we can see it is nothing but the spirilla, viral, helical shape or corkscrew form, and it is nothing but the vibrio type of arrangement. So each and every bacterial cell follow a particular shape and having a particular arrangement pattern. So by using that, we can differentiate the organism into different groups. So now, apart from the size and shape, bacteria also have a particular flagella arrangement pattern. So with respect to that, if we follow the flagella staining, we can carry it out, or by hanging dog method, we can determine whether they are motile or not. After that, we can stain for the flagella, and if we found a particular arrangement of flagella on a bacterial cell, we can found if the flagella, single flagella is present at the one end of the bacterial cell, it is nothing but the monotrichous type of flagella. If we found more than one flagella at a particular one end of the bacterial cell, we can call it as nothing but the Lophotrichous flagella. Apart from that, if we found a single or multiple flagella present on both the end of the bacterial cell, we can call it as nothing but the amphitrachous flagella or amphitrachous arrangement of flagella. If we found a multiple flagella present around the cell, we can call it as nothing but the peritracheous type of flagella. So with respect to the arrangement of flagella, we can differentiate bacteria into different groups. Now we'll talk about the pattern of endospore, or we can say spore arrangement of bacterial cell. All our bacteria are not spore former, but when we found the spore forming bacteria, the bacteria also follow a proper arrangement with respect to the endospore pattern. So if the spore is present in blue color, we can see it is nothing but the parent cell, and at the center there is a spore we can see, and if that present that endospore at the center of the cell, we call it as nothing but the central endospore. If we found that endospore present uh, somewhat towards the end of the bacterial cell, we called it as the subterminal endospore. And in case of that endospore present at the particular end of any one end of that bacterial cell, we can call it as nothing but the terminal endospore. So these are nothing but the different flagellar arrangement, bacterial cell, and shape. We can see when we observe that particular microscope bacterial cell under microscope, and if you follow the proper staining protocol for the bacterial cell. So by using that, we can differentiate the bacterial cell into different group on the basis of their staining pattern. So it is all about the morphological characteristics of bacterial cell. Another characteristics with respect to biochemical characteristics we'll discuss in next lecture.